हेलो हाँ संदीप 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 सर ने कर लिया ज्वाइन। हाँ वे सर
Very good afternoon to all of you present. Myself, Sherry, Assistant Professor in Department of Chemistry, DAV College, Abohar. It gives me immense pleasure to extend to you a very warm welcome with an object of encouraging the spirit of research and innovation. We feel proud to host a lecture on peptide-based nanomaterials as new aid scaffolds for bioimaging and biosensing agents. I extend my deep sense of gratitude to our honorable guest, Dr. Rohit Kumar Sharma, for sparing his valuable time for us. It is my great pleasure to introduce Dr. Rohit Kumar Sharma. He received his PhD in chemistry from National Institutes of Pharmaceutical Education and Research, Mohali, in 2009 and gained his postdoctoral experience from Nanyang Technical University, Singapore in 2011. Presently, he is working as an assistant professor in Department of Chemistry, Punjab University, Chandigarh. He published 43 articles in renowned journals and wrote a book on antimicrobial peptides. He has been granted 2 patents on his work and applied for 5 patents. He completed 3 projects related to peptides and nanoparticles. And moreover, 6 students have completed PhD under his guidance. And I also complete my PhD under his valuable guidance and support. Now I invite Dr. Rohit Kumar Sharma to share his research experience with us. Please keep your audio and video off during the webinar. Over to you, sir.
Yeah, thank you very much, Terry, for giving the opportunity.
Yes, sir.
Can you hear me?
Yes, sir.
Okay, fine. Thank you, Sherry, for giving this opportunity. And I would request you to kindly give me the sharing option so that I can share my screen.
सर रोहित सर आप पीसी और मोबाइल में से कोई एक चीज यूज कर लीजिए इसीलिए इको आ रही है
Okay, I think probably.
अच्छा
Yeah, I'm only using my PC. I'm not using any mobile.
सर आप पीसी और मोबाइल को एक को अनम्यूट कर दीजिए किसी एक को म्यूट कर दीजिए
I'm not using my mobile.
ओके ओके सर संदीप सर
Yeah. My mobile is off. I'm not using my mobile.
ओके सर
Yeah. I think you can probably
please start the
mute
session.
all the other audiences. I would request everyone to kindly mute so that there is no disturbance. Yeah. Okay. So I hope my screen is visible. Can I start?
You can start, sir.
Okay. Thank you. Okay. I would request everyone to kindly mute yourself. Today I will be presenting my work on peptide-based nanomaterials as new age scaffolds for bioimaging and biosensing agents. I think there is some mistake in the title. Kindly read it as biosensing agent. I think can everyone mute themselves? There are few people who are still unmuted. Okay, thank you. Can you hear me? Am I audible? Sherry, can you please unmute and let me know if I'm audible?
Yes, sir, you are audible.
Okay, thank you. And I hope my screen is also visible. So I shall be today talking about some of the applications that the chemistry or the organic chemistry people can do. I'll be discussing about those applications. And as Dr. Sherry has just introduced, it will be based on some biomolecules, especially the peptide-based materials and some nanomaterials, which are actually a new age materials. And as Sherry has just very kindly introduced me, I am from Department of Chemistry, Punjab University, Chandigarh. And so first, let me go take you through. Some of you have already visited our university probably, and also many of you must have visited Chandigarh. So I come from Punjab University, and it is situated in Chandigarh, and you can see that there are a lot of landscapes in Chandigarh. And so I generally present this slide to people who are outside the country, and so I thought just let me just introduce to you also. So this is a glimpse of Punjab University. Many of you must have visited it. This is these are some of the departments. Then there's a Gandhi Bhavan, and then there's a very renowned student center, which is a very famous place for visiting students and faculties also. So with these, I like to just give you an introduction of what I'm talking about. So we are actually working upon some new materials, or we are basically focusing upon some of the future materials which are actually now more towards organic compounds based materials, or we can say that the silicon which is already there in many of the mobile devices and other material devices, now it is getting converted to carbon, which is organic based devices. Now the carbon based materials are durable, they are economical, they are lightweight, they should be sustainable, and the most important thing with organic material is that they should be biodegradable. They are biodegradable. So I shall be talking about some of these organic materials which are derived from organic based systems, and we will be discussing about the applications as I move forward. And another thing that I want to introduce in the beginning is the nanomaterials. The nanomaterials basically are the materials which are of single unit small sized. Their size is generally between 1 to 100 nanometers, and there are many different type of nanomaterials which can be made. For example, starting from metallic nanoparticles, then going to ceramic nanoparticles, polymeric nanoparticles, carbon-based nanoparticles or nanomaterials. And the thing that I'm going to talk about is the bio-inspired nanomaterials. The word bio-inspired comes from the nature. So the nature has, whatever nature has made, we are trying to mimic that, and we are trying to make the bio-inspired or nature-inspired nanomaterials from those type of nanomaterials. So important thing is that for future nanomaterials or future materials, it is important that they should be economical, they should be durable, they should be lightweight, and of course This diagram here shows you the size index, how the nanomaterials can be ranged from the normal sizes. For example, a virus is, the coronavirus is right now going on. It is about 10 raised to power minus 7 meters, whereas nanomaterials are 10 raised to power minus 9 materials. So 10 raised to power minus 9 meters. So we are now going to talk about some of the materials which are actually very, very small in size. So when you mix those material, or when you make one monomer attached to another monomer, you then tend to get a bigger material. But the unit cell of these materials are very small sized, that is up to the nanoscale region. Uh, some of the bio-inspired future materials are actually derived from biomolecules which are present in our body. For example, proteins, DNA, lipid, carbohydrates. These are all molecules which are present in the body and in the future, which in the biological systems. And we shall be discussing about these materials. I will be introducing these biomolecules, and I will be talking about these basically these materials and their applications, which we will be taking up in the nanomaterials. So first and foremost, I'll be discussing about protein and peptides. You can make hydrogels from them. You can make nanofibrils, vesicles, spheres. Then there are nucleic acids. Nucleic acids are generally derived from RNA, DNA, and then there is a term called aptamer. There is origami based materials. Then lipids, which are very important for the cell walls of our cell membranes or cell walls of our cells. They can be changed into liposomes, neosomes, solid nanoparticles, cubosomes. And then there are carbohydrate materials, for example, cyclodextrine, cellulose, dextrine, chitosan. So all these materials actually now are termed in the combination with nanoscale materials as the future bio-inspired materials. So I'll be discussing about some of the applications of these. And then these applications include a lot of different opportunities. For example, in drug delivery, biosensor and diagnostics, medical implants, um, antimicrobial membranes, skin tissue repair, vascular grafts, scaffolds. So all these things are now actually being derived from these type of bio-inspired materials. The important point is that these bio-inspired materials are biocompatible. That is, they are actually the part of the body and they get assembled into the body quite easily. Then they are biodegradable. They can be easily degraded into the environmental system and don't cause any environmental problems. So they are environmental friendly also. They are economical because generally we get it from easily from the nature itself. And of course, for as a chemist, it is very important for us for them to be thermodynamically stable, and they have been found to be quite thermodynamically stable. So I shall be discussing about three things today. One application is in biomaterials. I shall be discussing about protein and peptide based biomaterials, that how we can derive new biomaterials from protein and peptide systems. Then I shall be discussing about drug delivery aspects, how these systems can be used in drug delivery and cell internalization. And then finally, I will be discussing about some of the sensors or some of the biosensors or bioprobes which are available. And I also shall be slightly discussing about how the coronavirus vaccine is coming up and how these biomolecules, talking about protein, peptides, nucleic acid, lipids, how these are playing a very important role in the vaccine development of coronavirus that is coming up against SARS-CoV-2 virus. So with these words, let me first introduce a bit of the topic. So probably you must have read in your textbooks that how the proteins are formed, and peptides are actually very small proteins. And generally, you tend to make bigger proteins by making smaller peptides. And so for formation of a peptide, amino acid should be combined together, and one carboxylic acid of an amino acid should be combined with the amino group of the another amino acid, and the condensation step happens, the water goes away, and this type of peptide is formed. So what is most important as from the chemistry point of view is that the carboxylic end of one amino acid should selectively combine with the amino group of the other amino acid. So you have to very judicially you have to um, combine or join together one carboxylic with the amino group of the other group, and then you tend to get this type of a carboxyl amide group, and for that you have to protect the other groups. So this is the general way of making a small peptide and then making a bigger protein. So with this, once the protein is formed, then it can take different shapes. For example, you must have read it can take a alpha helix shape, which is like a staircase, a round staircase going on through one axis, through the middle axis. And then there can be a beta pleated sheet, that is one sheet after another and then after another. So you can get these type of structures. Of course, apart from these structures, you can get some other structures also, but these type of combinations or these type of assemblies are very important for a protein to show its real nature. Yeah, whatever proteins are there in our body, they are assembled or have a special conformation. For example, some of them are alpha helix, or some of their parts are alpha helix and some of their parts are beta plated. So we have to understand these structures very carefully. Only then we can come to the applications of these proteins, and then we can talk about their applications. And many diseases, for example, Parkinson's disease, Alzheimer's disease, and these type of diseases are actually based on the denaturation of proteins in the body. So you have to understand these structures very, very carefully. So this is called as the secondary structure of the protein, and we can have different structures. Of course, primary structure is one when the protein gets combined. Then the secondary structure is one which I have just talked about, alpha helix or beta sheet or random conformation or other stuff. And when you have different secondary structures in one protein attached together, it forms a tertiary structure. Tertiary structure of a protein can have different secondary structures, and then different proteins actually combine together to form a self-assembled unit, which is consisting of different proteins. An example is a very good example is hemoglobin. You know that hemoglobin is a protein which is actually formed of 4 different proteins. So the quaternary structure of hemoglobin is having 4 different proteins which are attached together, and then another very good example is insulin. Insulin is also a small peptide or a protein which is a very important part of our body, and it plays a very important role in the body functioning. And this protein is also a tertiary structure which is having a lot of different secondary structures. So to understand these proteins or these biomolecules, you have to understand how these are actually combining different secondary structure and forming a bigger tertiary structure. So with these background, I'll go to some of the applications of these things. So if in case some of you have a problem with the language, I can also speak in Hindi, and I can also explain the things in Hindi, or even if Punjabi is also fine with me. So please let me know if you can write it in the chat box if you find the language problem. Okay. So what are the forces that stabilizes the protein? As a chemistry student, it is very important to understand how these proteins are stabilized. Proteins have covalent bonds, as we have discussed, they form amide bonds. Apart from covalent bonds, there are hydrogen bonds which are there between different units. They also tend to stabilize the protein structure. Then there might be some metal ligands. For example, zinc is very well known, which actually combines together and forms a very good complex with histidine in the protein. And then there are ionic interactions, then there are disulfide bonds, and then there are non-bonded interactions. So these type of structures are there, or interactions are there, which actually stabilizes the protein and helps the protein to do the biological function. And all these are actually chemistry terms. Only a chemist like us can understand these things, and we can make it explain it better to the biologist. So don't think that these are the things which are beyond your concepts or beyond your reach. The chemists are doing wonderfully well in these fields, and there has been a reportage of getting Nobel Prizes also from the biological streams to the chemists. So I'll encourage students to kindly go into this, look into this, and you can make career into these fields also. Okay, so with this background, I'll be discussing about some of the applications of biomaterials, that how we have used these biomolecules to make new type of biomaterials. What we have done in our lab is initially we have started with small biomolecules such as amino acids and small peptides, and we have tried to assemble them into different supramolecular architects. trying to form different forms. For example, when the linear form was formed, we made some fibers. Then tubes were made from these small biomolecules. Then we have made some orthotropic forms in which we have tried to make particles. We have tried to make matrices, and then we have tried to also make different membranes. So we are mainly focusing upon small peptides or small amino acids, and we have tried to make self-assembled structures out of this. It's a very new topic. There are very few reports available on these type of topic, and it is about 10 to 15 years old field, and there are a lot of opportunities which are there in this field also in future. So what we have focused upon is we have used amino acids, as I have discussed. We have used small peptides. Then we have also used some chemically modified amino acids and peptides, and we have tried to assemble them to form different type of units like nanofibrils, sheets, tubes, and different type of, and then we have tried to see what are the applications of this. From the chemistry point of view, it is very important to understand that you need to have a particular solvent system and particular conditions in which these type of materials can be combined together to form different different forms of these materials. And then of course, what are the chemical functionalities? For example, if you have a cationic moiety, this generally combines electrostatically with a negatively charged moiety, and then it tries to self-assemble and give different structures. And of course, as we have discussed in the case of proteins, you need to see what are the non-covalent interactions, what are the hydrogen bonded bonds are there, what are the ionic interactions, how these interactions are playing part, and then you tend to get different structures. So I'd like to just start with one of the reports that we have published recently, in which we worked upon some aromatic amino acids. There are three or four aromatic amino acids such as phenylalanine, histidine, tryptophan, tyrosine. There was a report in the journal Science in which it was discussed that the proteins in the body actually degrade into smaller amino acids, and these amino acids self-assemble in the body, and then they tend to aggregate in the body, and because of that lot of problems occur. For example, in the case of Parkinson's, it was found that it is smaller peptides or amino acids which self-assemble, and they stop the functioning of a normal protein in the body, and because of that there are a lot of complications. So what we did was we took smaller amino acids, we tried to solubilize them in different solvent systems, but we found that only the aromatic amino acids were getting solubilized into those particular solvent systems, and they were forming different self-assembled structures. So when we dissolve this phenylalanine and we try to see that how it is functioning, we found that this phenylalanine was making very nice nanofibrils. Histidine was also trying to make some pseudo sheets, as well as tryptophan was making some floral designs, and tyrosine was making needle-like structures. So you understand, when these amino acids are present in the body and they get degraded in the body, they can also self-assemble and make these type of structures, and these structures can then cause problems. So what happens is, when these self-assembled structures are formed, and when these self-assembled structures have a particular toxicity, and that toxicity is actually measured by attaching a dye with it and see whether they cause amyloidosis. There is a concept which is called as amyloid generation. When these amyloids are formed, they generally tend to form a very stable structure, and then they don't degenerate in the body, and then they stop a lot of biological functions. So to make this understandable, मैं हिंदी में समझाना चाहूँगा स्टूडेंट्स को कि जब हमारी बॉडी में जो प्रोटीन्स होते हैं वो जब ब्रेक कर जाते हैं, तो वो ब्रेक करके दे फॉर्म डिफरेंट सेल्फ असेंबल्ड स्ट्रक्चर्स. अब एक स्ट्रक्चर जो होता है वो फॉर्मेशन होती है एमिलॉइडिक स्ट्रक्चर की. अगर वो एमिलॉइडिक स्ट्रक्चर बन जाए तो वो ब्रेक नहीं करता है बॉडी में, और वो ब्रेक नहीं करने की वजह से देयर आर लॉट ऑफ कॉम्प्लिकेशंस हो जाती हैं बॉडी में, और उसकी वजह से बहुत सारी बीमारियां हो सकती हैं. तो understanding of self-assembly of metabolites such as these amino acids is very important to help in the treatment of very important pathological diseases. तो हमने क्या किया, हमने ये amino acid लिए, we tried to make them self-assemble in the lab conditions, and we saw that what structures they are forming, and we did some NMR studies, XRD study, TGA studies, सब इनको understand करने के लिए FTIR, फिर हमने fluorescence के साथ we have attached a dye with it and we try to see whether the amyloidic structures are forming. So जब हमने डाई को लगाया इन सेल्फ असेंबल स्ट्रक्चर के साथ, फॉर एग्जांपल when we attached the thioflavin T dye, we found that many of them were forming amyloidic structures. So this is a very important inference that we found कि जब हम इन small amino acids का self assembled structure बनाते हैं, this was the first report which has come so far. So उसके अंदर amyloidic structures form होते हैं lab conditions में. अगर lab conditions में these are forming, these can also be checked in the in vivo system या body के अंदर भी हम इसे check कर सकते हैं. So these, if this is true, then this is one of the major reasons that many pathological diseases are happening. So for treating these diseases, we have to stop these amyloidic structures. So that is the work, that is what we are trying to do with some of our collaborators to see कि biologists के साथ मिलके हम check करना चाह रहे हैं कि what are the possibilities to stop these type of amyloidic structures. But this is what we have done in our lab to understand the aromatic amino acid self-assembly. Okay. So this was just one example. इसी के साथ हमने we have done some other things in which we have taken smaller peptides. For example, we took a dipeptide. Dipeptide जो होता है उसमें दो अमीनो एसिड जुड़े होते हैं, एक ट्रिप्टोफेन and one phenylalanine. This is a dipeptide which was formed. उसके साथ कुछ protecting group भी लगे हुए थे. For example, FMOC on the one side and ester group on the other side. So we have tried to self-assemble these structures and we have tried to form nanospheres. So these very clear nanospheres are formed. Now, if you understand that if you can make a nanosphere from a biodegradable, environment-friendly material, for example, protein and a peptide, you can actually put a different type of a drug or a foreign moiety, and you can take that moiety inside the body system. So what we have done is we have made these nanospheres from the peptide monomers. We have then introduced a drug molecule into this sphere, and we have then checked whether these drug molecules are going inside the body. And for this, we actually added two different type of drugs. One was antibacterial, which is ampicillin, and one was anti-cancer drug, which was doxorubicin. So that drug was actually put inside the nanosphere, which was made from a small dipeptide system, and everything is chemistry. There is no biology here. So we have tried to inject these drugs, and we have seen that these drugs are very nicely getting internalized into the biological system or cells. And for agar ab aap samajh sakte ho, once you are taking a drug or a medicine, if that medicine is now targeted and it is protected, you need very less quantity of that drug or of medicine. So the efficacy of the drug actually increases a lot with these type of applications. So you can actually do this. There are a lot of drug delivery applications apart from this type of things also, but this is one of the very latest reports in which peptide based nanospheres have been formed and the drug molecule has been introduced. So we calculated, we found that for ampicillin, more than 50% of the ampicillin was getting encapsulated, and about 80% doxorubicin was getting encapsulated. So with this work, we have actually finalized that we can actually, now we're trying to do some in vivo studies. We're trying to see that how this can be used for in the body systems directly. Another work that we have done is we have taken small amino acids. I'm actually presenting this as new materials. There are bio-inspired materials which can be found in different ways. So what we have done is phenylalanine is an amino acid. We have tried to self-assemble a phenylalanine, as I've shown in my earlier slides. Phenylalanine forms a lot of nanofibrils like this. So nanofibrils are formed like this. We have tried to see that what is the unit cell. You understand from X-ray that there is a unit cell, the one particular monomer. What is the monomer of this nanofibril? What we have found is that small phenylalanine system dimer is formed, which actually gets entangled together using different hydrogen bonds or ionic interactions, and these dimers then join together to form a nanofibril system. So this is also a latest work that we have presented, and in which we have tried to show that these type of nanofibrils which are formed are actually formed from small dimer systems from the phenylalanine. And in this we have used a latest technique which is called as small angle X-ray scattering study or SAX study, in which network of a nanofibril system. And this was actually highlighted on the back cover of Soft Matter Journal, which is a very renowned journal in the material chemistry. And this is the first report in which we have tried to show that these nanofibrils are formed from small units of dimers of phenylalanine. So this is about the bio-inspired materials, that how these bio-inspired materials, new type of bio-inspired materials are going to be the future of our material chemistry or material sciences. So with this, I'll now move to another type of application in which we have tried to remove some. For example, this is one of the small dipeptides that we have synthesized in the lab, and then we self-assembled. We found that these dipeptides are forming a lot of different type of structures. For example, some are forming this floral structure, some are forming these needles, and these needles are quite stable, and these are characterized using scanning electron microscopy, which is called as SEM, or some of them have also been characterized using transmission electron microscopy. So we have tried to generate different self-assembled systems, and we found that these self-assembled systems are showing very good application in dye removal. Specifically, cationic dyes are actually getting attached to these self-assembled peptidic systems, and they are getting removed. For example, crystal violet is a cationic dye which got removed when it was attached to these type of nano systems made from different peptides. And we could show that the maximum adsorption capacity was quite good, about 625 milligram per gram, which is very, very good as compared to already available systems. And we are now in the process of actually taking it and making it at a larger scale also. So we are also working up, apart from peptides, there is also a term called as peptoid. Peptoids are those peptide-like systems in which amino acid is not used, but normal organic molecules are used to couple to one another, in which the side chain of the amino acid is not attached to carbon, but to the nitrogen. So we have synthesized different peptoids, and these peptoids are novel peptoids, and we are trying to see different applications, and we found that these peptoids are also showing very good applications in the removal of cationic dye. For example, they are also forming different self-assembled structures like needles, floral submolecular units. Then there are some branch chains and other stuff, and they are also specifically working upon cationic dyes. And these peptoids are right now, we are in the process of submitting this manuscript also. So this is an idea about what are the organic based materials or future materials that are coming up slowly. The second application that I'm going to talk about is cell internalization, how these materials can be used to go into the biological system, how you can actually add into the biological system. For that, you have to understand a term called as nanoconjugate. Nanoconjugate is made up of two words. One is nano, another is conjugate. So when you make a nano material or a nanoparticle system, and then you attach some moiety with it, or you conjugate some moiety with it, then it is called as nanoconjugates. And we know that nanoparticles are generally ranging from 1 nanometer to 100 nanometer, and their surface can be positively charged or negatively charged, and this surface can be attached to different type of materials such as drugs, dendrimers, liposomes. These are all conjugates that you can actually attach with the nanosystem. And these nanosystems can have different shapes. For example, they can have sphere shape, cube shape, rod, plate, and other type of shapes. So you can have this type of a nanoconjugate system, which can be used for different applications. What is important from the chemistry point of view is that you should understand that what type of chemistry it is there. Generally, we work upon, like, for example, if we work upon gold nanoparticle system, then gold sulfur interaction is very important, which actually leads to this type of nanoconjugate. Then there can be a specific enzymatic interactions. Then there can be diffusion based functionalization, or then there can be an electrostatic interaction, direct electrostatic interaction, which is possible. So this type of interactions you have to understand, and then you have to build your nanoconjugate system. So we worked upon amino acid capped gold and silver nano hybrids. We made some gold nanoparticles. We conjugated some amino acids with it. We found that some of the amino acids were really helping the gold nanoparticles to form. Similarly, we also made some silver nanoparticles and found that some of the amino acids are playing very important role. This is an figure actually shows that some of the amino acids are playing very important role. The purple color in here actually shows the reduction by the amino acid, whereas the yellow color is the yellow color is the efficient reduction, purple color is inefficient reduction, and gray color is no reduction. Similarly, in the gold nanoparticles, some of them showed very good examples of conjugating, whereas some were not showing good examples. So what I'm trying to tell you is that you can make nanoparticle system or the nanomaterial systems directly from the bio-inspired materials such as amino acids and peptides. So this was an example with amino acids. We also conjugated some peptides with these gold nano silver hybrids, and these peptides were then found to be these systems were found to be quite stable. And this is a transmission electron microscopy images of these nanoparticles, which are showing quite well monodispersed and stable nanoparticle systems that were found. So we were happy that we got this. We also quantified that what is the quantity of the amino acid or the conjugating system like peptide is required, and we have just reported it recently in the Nature Scientific Reports. So with this, we were actually interested in making some of these systems work for cell internalization. So the idea that we discussed was that if we have a gold nanoparticle system and if we conjugate it with a specific cell penetrating peptides. Now these cell penetrating peptides are specific for bacteria or some other microbes which can go directly into those microbial systems. So then we can have these type of conjugates in which gold nanoparticle is conjugated with cell penetrating peptide. And if we then add a fluorophore, then we can introduce this complex, this conjugate, which is a gold nanoparticle cell penetrating peptide system into the microbial system, and then we can understand. So this is all chemistry. What we require is to understand how to make these gold nanoparticles, how to make the conjugates, and then how to do the application. So we, with the help of some of our collaborators, we tried to introduce these into different cells, but what we found was that there was not very efficient internalization. You can see that there are signals coming in the cells, but there are very few signals that are coming in the cells. And this was not very efficient. What we found was probably in the earlier slide when we added the FITC here, additional FITC here, that has actually changed the conformation of this conjugate. Now, once the conjugate conformation is changed, it is difficult to internalize into the bacterial cell directly. So we changed the scheme. We used, instead of gold nanoparticle, we used quantum dots. Now the quantum dots are semiconductor nanoparticles. They are biocompatible and very highly fluorescent, and you don't need any extra tag. You don't need to add any fluorophore into it. You can directly use the quantum dot, and that helps it to directly go into the system, and that will not change the conformation of the conjugate. So we synthesized the peptide, we made the conjugate, we made this different conjugate in which quantum dot was attached to the cell penetrating peptide, and then we try to see whether it is going into the bacterial cell. And for our liking, we were happy to see that there was a very high internalization in the case of cell penetrating peptide and quantum dot conjugates attached with the drug molecule also. And this paper has been recently published in ACS Applied Biomaterials, and this is again one of the first reports in which quantum dot based peptide systems have been used for cell internalization. So what I'm trying to tell you is that you can actually use your chemistry knowledge to do a lot of applications in the biological system also, if you understand both of these things. Okay, so before I move forward, I'll just now like to go back to basics. So I discussed about peptides. Now I'll discuss about nucleotides, or what you call as DNA RNA structures. So DNA RNA or nucleotide structure is composed of three parts, as you might be knowing. One is phosphate, another is sugar, which can be ribose if it is RNA, it can be deoxyribose if it is DNA. And then there are two types of bases, purines or pyrimidines, depending upon which type of system you're using. And all three actually combine together to call as nucleotide. So for example, RNA is a nucleic acid, which is a small part of a nucleotide, and you that in DNA, as I have discussed, sugar is different and the different bases are used. So this, if in the case of DNA, instead of hydroxyl at the 2 position of ribose, if you have a hydrogen here, it is called as deoxyribose. So that is the only difference between RNA and DNA as far as sugar is concerned. Of course, in RNA you have a uracil, in DNA you have a thymine as a base. So this is another difference that is there. Otherwise, they're quite similar. So in DNA, we all know DNA has a double helical structure, and there are minor grooves, there are major grooves, and these are actually trying to form a double helix structure in which two strands of DNA are joined together by hydrogen bonding between different base pairs. So what I'm trying to explain to you is, because I'm now coming to some of the vaccines that are being used for coronavirus, they are actually based on these biomolecules. So what nature has proposed, nature has chosen proteins, DNA, RNA, and all these type of materials as their molecules. So whatever our body is made up of these type of materials. DNA is the main parent compound which goes from one generation to another generation. From that the protein comes up, from that the carbohydrate, lipids, everything comes up from there. So nature has chosen this. So we as human beings, as scientists, are also now going back to nature. We are also trying to find out the solution in the nature. So similarly, there is a term called as lipid, which is also a natural material or material made from the nature. These are very important as far as the cell walls or the cell membranes of our cells are concerned. They are basically hydrophobic molecules and they contain carbon, hydrogen, oxygen. They are also typical organic molecules. Generally, we have glycerol based lipids in which they are made. Glycerol is you know that it is a tri-hydroxy based compound in which if you remove the two hydroxy groups and you attach some hydrophobic unit, you tend to make lipids made from the glycerol unit. So there are different types of lipids. For example, fats, phospholipids. Phospholipids are very important because they form the cell membranes. Then oils, waxes, steroid hormones, triglycerides, all these are lipid-based molecules. Phospholipids, as I've discussed, they are structurally and chemically similar to triglycerides. One fatty acid of the glycerol is replaced with the phosphate group. That is why it is called as phospholipids. So phosphate group is hydrophilic, whereas other units are hydrophobic. So these, why I'm explaining to these natural molecules to you is because I want to explain to you the coronavirus virus. What is this coronavirus actually? This is not a fully living species. This is something like a partial living species. So this coronavirus is actually made of these biomolecules. It is only a combination of these biomolecules. There are spike glycoproteins on its surface. Glycoproteins come from one part from the carbohydrates, that is the glycan part, and another part from the proteins. So these are the glycoproteins. And then there are membrane proteins, which are composing of its membrane. Then there are nucleoproteins inside. Then there is a genomic RNA. RNA is a nucleotide. So this is the structure of the virus. So what is this? This is actually a combination of a different biomolecules which has a tendency to infect one part of our body, one or some other parts of our body, and we don't have any particular medicine or a vaccine against it so far. So what is the solution? How the vaccine will come for this virus? So this will come, there was a recent report from Nature that there are 4 different types of virus vaccines which are working upon, the scientists are working upon. One of this is based on nucleic acids. You are making different DNA, RNA based vaccines. Another one is protein based vaccine. You're taking different proteins or peptides or protein subunits for making these vaccines. And then there are viral vectors. There are vectors which are replicating and non-replicating. You are trying to use them. And of course, then there are virus-based vaccines also. You're trying to use different viruses itself, which are activated or weakened. So what you have to understand is the answer to this problem of coronavirus will also come from chemistry or the medicinal chemistry. That once you understand the structure, then you can synthesize this. And everything that is being done to synthesize is based upon the organic chemistry protocols. So you have to understand chemistry only, then you will make this virus. Of course, once you make that vaccine, you have to test them, you have to see how these function and how these, so then you need biologists. Not only biologists, you need pharmacologists, you need medical doctors and other stuff. But before you start anything, you have to synthesize this, and there the role of a chemist or a specifically organic chemist is very very important and very very vaccine making or medicinal chemistry or these type of studies. Okay, so with this background, I'll come to the last part of my talk, which is called as applications in biosensors. So we have tried to use different type of materials to make different biosensors also. So what we worked upon was we make these biomolecules, conjugated nanomaterials, which are based biosensors. For initially, we tried to see that how we can there is a huge problem of pesticides, specifically in your area, in the Malwa area of Punjab. You know that the water is having a lot of pesticide problems. You have to detect the pesticide. The problem with the detection of pesticide is that you have to take them to lab. You have to use very sophisticated instrumentations like HPLC, GCMS, and others. And you need to have a skilled manpower. It is a very time-consuming process. So what we need is we need a on-site detection of pesticide. For example, a farmer, किसान अपने खेत में जाके डायरेक्टली पेस्टिसाइड को डिटेक्ट कर सके, ऐसा कुछ चाहिए हमें जो कि डायरेक्टली हो सके. So for that we actually worked upon this biomolecule based nanomaterials, and initially we worked upon the colorimetric detection of 4,8. 4,8 is a organophosphate pesticide, and we made some gold nanoparticles, and we made a aptamer. Aptamer is a single stranded RNA unit. एप्टामर जो होता है, नाम से नहीं घबराना, नाम उसका कुछ भी हो, बट उसका सिंपल है कि वो आरएनए का एक स्ट्रक्चर है which has a specificity which can directly interact with that pesticide. उसके साथ वो directly pesticide के साथ attach कर सकता है. So what we did, we synthesized gold nanoparticles, conjugated the aptamer with that gold nanoparticle to make this type of a complex. Then when we added the 4,8, जब हमने pesticide डाला उसमें, तो pesticide क्योंकि aptamer के लिए specific था, that pesticide took away the aptamer and the gold nanoparticle was free. उसके अंदर जब मैंने इलेक्ट्रोलाइट डाला, NaCl सिंपल सॉल्वेंट डाला, तो जब हम इलेक्ट्रोलाइट डालते हैं तो उसमें क्या होता है, पॉजिटिवली चार्ज सोडियम है, नेगेटिवली चार्ज क्लोराइड ग्रुप है, तो वो नेगेटिवली चार्ज पॉजिटिवली चार्ज जो है गोल्ड के सरफेस पे जाके गोल्ड के सरफेस के जाके इंटरेक्ट कर जाते हैं और गोल्ड को एग्रीगेट कर देते हैं. अब जब गोल्ड एग्रीगेट हो जाता है तब उसका कलर चेंज हो जाता है, वो ब्लू हो जाता है. नॉर्मल गोल्ड जो होता है वो रेड कलर का होता है, गोल्ड नैनोपार्टिकल्स. सो व्हाट वी डिड, जब हमने 4,8 डाला उसके अंदर, तो उसने एप्टामर को साइड पे ले गया, गोल्ड नैनोपार्टिकल फ्री हो गए. उसके अंदर जैसे ही इलेक्ट्रोलाइट डाला तो वो ब्लू कलर हो गया. ऑन द अदर हैंड जब हमने 4,8 नहीं डाला था तो वो कॉन्जुगेट ऐसे का ऐसा रहा. अब क्योंकि एप्टामर स्टेबलाइज कर रहा है गोल्ड नैनोपार्टिकल को, तो इलेक्ट्रोलाइट के आने की वजह से कोई भी चेंज नहीं आया और गोल्ड जो है रेड कलर का रहा. तो ये आप कलर देख रहे हैं ना, you're seeing these colors, these are actual colors. The color was red when there was no 4,8 present. 4,8 जो है पेस्टिसाइड, और कलर ब्लू हो गया जब 4,8 आ गया. इसकी सारी ऑप्टिमाइजेशन की और चेक किया. तो आप क्या कर सकते हैं, आप डायरेक्टली जाकर चेक कर सकते हैं. अब उसके ऊपर you can take the pesticide, you can take the gold nanoparticle and you can check what is the color change, what is happening to the directly. आप अपनी किसान जो है अपने खेत में ही इसको कर सकता है, तो इमीडिएटली वो चेक कर सकता है कि उसके अंदर पेस्टिसाइड प्रेजेंट है या नहीं है. और हमने चेक किया कि आप नैनोमोलर रीजन तक इसको चेक कर सकते हैं, नैनोमोलर रेंज तक इसको चेक कर सकते हो. दैट इज अ वेरी सेंसिटिव टेक्निक, बहुत ही सेंसिटिव टेक्निक है, एंड दिस वाज पब्लिश्ड. एंड उसके बाद वी वर्कड अपॉन अनदर पेस्टिसाइड व्हिच इज कॉल्ड एज मेलाथाइन. इस केस में भी वी यूज पॉलीमर, एप्टामर एंड गोल्ड नैनोपार्टिकल्स. सो इसमें हमने एप्टामर लिया, उसके साथ जब मेलाथाइन लगाया तो मेलाथाइन ने एप्टामर के साथ इंटरेक्ट करके इस तरह का एक कॉन्जुगेट बना लिया. उसमें जब पॉजिटिवली चार्ज हमने पॉलीमर लिया तो क्योंकि वो ऑलरेडी इंटरेक्टेड था तो उसका कोई इफेक्ट नहीं हुआ इस कॉन्जुगेट के ऊपर. एंड देन वी फाउंड दैट दिस इज रिमेनिंग फ्री. उसके अंदर जब गोल्ड नैनोपार्टिकल डाले नेगेटिवली चार्ज, तो नेगेटिवली चार्ज गोल्ड नैनोपार्टिकल पॉजिटिवली चार्ज पॉलीमर के साथ फिर इंटरेक्ट कर गए और गोल्ड नैनोपार्टिकल एग्रीगेट करके उनका कलर ब्लू हो गया. ऑन द अदर हैंड जब हमने नहीं लिया था मेलाथाइन तो एप्टामर फ्री रहा. एप्टामर अब नेगेटिवली चार्ज है और पॉलीमर पॉजिटिवली चार्ज है, तो उसने अपना एक कॉम्प्लेक्स बना लिया इस तरह का. और जब हमने गोल्ड नैनो पार्टिकल डाला तो वो उसके साथ इंटरेक्ट नहीं किया और वो रेड कलर ही रहा. सो इंपॉर्टेंट पॉइंट ये है कि जब आपने पेस्टिसाइड डाला, when you put the pesticide in this, the color changed to blue, the gold nanoparticle, and when the pesticide is not there, the color remained red. And in this case we found the लिमिट ऑफ डिटेक्शन जो थी वो पिको मोलर रीजन तक 10⁻⁻12 मोलर रीजन तक रही. काफी सेंसिटिव डिटेक्शन टेक्निक है और आप इसको डायरेक्टली यूज कर सकते हो. और दिस वाज पब्लिश्ड इन Biosensor and Bioelectronics. The impact factor of this paper is more than 10.6 or 10.7, something. That is the impact factor right now. So it's very renowned paper, and this work was taken very well. तो हमने उसके बाद we worked upon another scheme. Instead same scheme को हमने यूज किया है, पॉलीमर की जगह पे पेप्टाइड यूज किया, बायोमोलिकुलर बेस्ड टेक्निक. इसमें this is the first technique in which peptide was used for this type of biosensing techniques, and this was published in Chemical Engineering Journal, or this was quite well taken. उसके Chemical Engineering Journal के first page के ऊपर आया था ये, so on the cover page of it. ये impact factor पुराना है 8.35, अब इसका impact factor 10.3 है, so that impact factor has changed. So this was very well taken, और cover page पे आना journal के, it is quite a big thing, and this was taken quite well. So what I'm trying to tell you is that you can use your chemistry knowledge to do a lot of applications. And abhi ka kya hai scenario, that you need to have applications, then only you will get funding. Otherwise you will not get funding. So you have to work upon different applications. Similarly, so we worked upon with a German group, our collaborator in Germany, who are a polymer chemistry based journal. We worked upon quantum dot and polymer based technique for malathion detection, which was also published in Biosensor and Bioelectronics. And we found that very good biosensing technique was approached. Recently we published in Chemical Communication, in which we actually pesticide ke alawa we have also worked upon metal ions. For example, we have checked the detection of lead. We have used the same type of technique, jaise ki aptamer liya. When lead is present, it forms a complex. For that, aptamer now is specific to the lead in this case. फिर जब पॉलीमर डाला तो पॉलीमर फ्री रहा और गोल्ड नैनो पार्टिकल को कॉन्जुगेट कर दिया, एंड देन द कलर चेंज्ड. And the limit of detection for lead was 98.7 picomolar, so very high sensitivity. And this was published in Chemical Communication recently. We have just published a paper. This is actually published already. This is in Talenta, where we have worked upon biometallic nanocluster system. Nanocluster क्या होते हैं? These are also very highly fluorescent type of molecules. In nanoparticles से थोड़े different होते हैं. In this we are getting very high fluorescence. ठीक है, नैनो पार्टिकल जनरली don't have fluorescence. So in this we have worked upon biometallic nanoparticle systems like gold and silver nanocluster systems, or we have tried to see the detection of different pesticides also in this case, and this has also been taken quite well. Right now we are working with a group in IIT Madras, in which we are trying to make a small kit type of a thing. You know that you can actually make a kit, like for example for pregnancy kits are there, you can actually see the color change if the analyte is present or not present. So we're working upon that kit scheme for these type of pesticide systems, and this was a project that we are working upon these days. So I have tried to explain to you peptide based biomaterials कैसे हम बना सकते हैं, कैसे उसकी एप्लीकेशन कर सकते हैं, then nano conjugation based by drug delivery and cell internalization systems, then sensing schemes using different bioprobes. So all these things are actually chemistry based techniques. There are two different parts. One is the nanomaterial part, and then other is the biomolecule thing. So we are working upon different applications, or we have tried to understand that what is the relevance in the diseases, then how the biocompatible and biodegradable drug delivery systems work, and then how peptide and aptamer based biosensing. And I've also tried to just explain to you in between that how this coronavirus vaccine is coming up. They are also coming up mainly from these type of biomolecules. So with this, I'd like to thank Afos. You have your own faculty, Dr. Shari Mittal. She is actually my first PhD student. So I shall always be proud of her in that sense. And of course, then there are other students. I have tried to explain to you, work of these students, the students whose colors are in red or maroon color. So there are other students also worked upon with me. There are MSc students who do projects. Sorry, I cannot write the name because there are every year I get 2 or 3 MSc students. And of course, I like to thank the collaborators. Collaborators not only from PU but from abroad. We have collaborations in Germany, Japan specifically, and then there are some collaborations from IITs. And last but not the least, I like to thank the funding agencies. They have been really generous on us to give us different fundings. So with this, I thank you, and in case you want to ask any questions, I shall be happy to take them. Thank you very much.
Thank you, sir. Any query you can ask directly from the sir, or you can write in the chat box.
Yeah, I hope I was understandable, because there are some things which are slightly complex because this is all application based work. But I shall be happy if you want to ask anything. This is all chemistry work. We are synthesizing different biomolecules, we are synthesizing different nanomaterials, and then we are trying to see their applications. And specifically for students who are actually trying to pursue their careers in research, this is a very you can actually, you should look at your fields in which you can do a lot of applications, because the jobs in future are going to be coming in only mainly in the application derived fields. I'm not talking about teaching and these type of jobs. I'm talking about industrial jobs, and then you can actually start a lot of there are a lot of startups also coming up in these fields, where you can actually make different type of products which are having applications in day-to-day life. So this is all what we are trying to do. We're trying to make things applicable in day-to-day life. Yes, you can ask me in any don't be shy. अगर आपको हिंदी में पूछना है हिंदी में पूछ सकते हो, पंजाबी में पूछना है तो भी पूछ सकते हो. I can talk in Punjabi also. Yes. Or you can just send your questions to Dr. Sherry. She can pass it on to me.
Sir, there is one question by a student Chirag. Please elaborate aptamer more.
Okay. So aptamer is just an aptamer एक नाम है. इसमें कुछ घबराने की बात नहीं है. जिससे आपने डीएनए आरएनए तो पढ़ा ना, तो एप्टामर जो होते हैं वो डीएनए या आरएनए होते हैं. जनरली हम जो यूज करते हैं वो आरएनए बेस्ड एप्टामर है. जैसे मैंने बताया आपको कि आरएनए या डीएनए जो है वो एक 3 चीजों से बना हुआ है, एज ये फॉस्फेट, शुगर और बेस. तो ये भी जो एक एप्टामर है वो आरएनए है या डीएनए है जो कि स्पेसिफिक होता है. हम क्या करते हैं, हम वो वाला डीएनए या आरएनए लेते हैं. फॉर एग्जांपल अगर हम पेस्टिसाइड की डिटेक्शन कर रहे हैं, लेट्स से मैं मेलाथाइन की डिटेक्शन कर रहा हूं, तो मैं मेलाथाइन के साथ जो इंटरेक्ट करेगा सबसे अच्छा डीएनए या आरएनए, उसको मैं लेता हूं और उसको मैं एप्टामर बोल देता हूं, बिकॉज वो एक स्पेसिफिकली मतलब उसकी जो कॉम्बिनेशन है उस एनालाइट के साथ वो एप्ट हो रही है, एंड इट इज अ मोनोमर. सो वी आर ट्राइंग टू नेम एप्ट monomer conjugation. So the word is coming from there, that aptamer conjugation is taking place with that particular analyte. तो हमारे पास बहुत तरह के एप्टामर हो सकते हैं या बहुत तरह के आरएनए हो सकते हैं, लेकिन जो सबसे अच्छा उसके साथ इंटरेक्ट कर रहा है वो हम पहले चेक करते हैं, दैट व्हाट इज द इंटरेक्शन ऑफ दैट आरएनए और डीएनए विद दैट एनालाइट. फिर उसको हम सिलेक्ट करके उसको हम अपनी स्कीम में यूज करते हैं. सो इन ब्रीफ, aptamer is just a small RNA DNA unit which is showing very high specificity या combination specificity to that particular analyte. जो प्रोटीन भी हो सकता है, वो पेस्टिसाइड भी हो सकता है, वो लेड आयन भी हो सकता है, मेटल आयन भी हो सकता है, कुछ भी हो सकता है. So aptamer is a nucleotide, DNA or RNA which is having specific interaction. I hope I could understand. I can make you understand this.
Next question is, how peptide conjugate nanoparticle interact with cell membrane of bacteria?
Okay, that's a very good question. सो सेल, अब मैंने जो बात की, मैंने आपको बताया, लेट मी गो बैक टू दैट। जैसे ये एक स्कीम है, मैंने आपको, आई हैव टोल्ड यू कि वी कैन हैव नैनो पार्टिकल्स, फॉर एग्जांपल गोल्ड नैनो पार्टिकल, या बाद में हमने क्वांटम डॉट बनाए, क्वांटम डॉट भी नैनो पार्टिकल ही होते हैं। तो जब मैंने ये गोल्ड नैनो पार्टिकल के साथ सेल पेनिट्रेटिंग पेप्टाइड लगा, अब ये पेप्टाइड की खासियत है, ये पेप्टाइड ऑलरेडी लिटरेचर में अवेलेबल हैं कि जो एक पर्टिकुलर सेल को ज्यादा इंटरेक्ट करते हैं। फॉर एग्जांपल बैक्टीरिया को ज्यादा इंटरेक्ट करते हैं, हमारी बॉडी के जो सेल्स हैं ह्यूमन सेल्स उनको इंटरेक्ट नहीं करते। उसके पीछे कारण है, देयर इज अ रीजन फॉर दैट। जो बैक्टीरियल सेल मेम्ब्रेन होती है दैट इज नेगेटिवली चार्ज, वो नेगेटिवली चार्ज ज्यादा होती है। उसके अंदर डिफरेंट तरह के लिपिड्स होते हैं, उसके अंदर ज्यादा फॉस्फोलिपिड्स होते हैं, जैसे मैंने बात की ना आपको फॉस्फोलिपिड्स की, उसके अंदर ज्यादा फॉस्फोलिपिड्स होते हैं जो कि नेगेटिवली चार्ज होते हैं। लेकिन हमारी बॉडी के अंदर स्फिंगोलिपिड्स ज्यादा होते हैं, दीज आर न्यूट्रल लिपिड्स, रिलेटिवली न्यूट्रल लिपिड्स, उतने ज्यादा नेगेटिवली चार्ज नहीं होते। तो जो हमारा पेप्टाइड अगर हम ज्यादा पॉजिटिवली चार्ज लेंगे या ऐसा लेंगे जो कि बैक्टीरियल सेल के साथ ज्यादा अच्छा इंटरेक्ट कर सकता है और हमारे साथ अच्छा इंटरेक्ट नहीं कर सकता, तो उस पेप्टाइड को अगर मैं गोल्ड के साथ उसके सरफेस के ऊपर कॉन्जुगेट कर दूंगा, ठीक है, ये सब केमिस्ट्री की टेक्निक्स हैं, कैसे आप कॉन्जुगेट करते हो, कैसे वो गोल्ड नैनो पार्टिकल के साथ अटैच होता है। तो अब क्या होगा, अब ये जो सेल पेनिट्रेटिंग पेप्टाइड उसने अब लगा दिया, वो आपकी बॉडी के अंदर, आप मान लो आपके अंदर बैक्टीरियल इन्फेक्शन हो जाता है बॉडी में, तो वो हमारे सेल्स के अंदर नहीं जाएंगे क्योंकि वो हमारे सेल्स के साथ इंटरेक्ट नहीं कर सकते, बट वो बैक्टीरिया के सेल्स के अंदर ज्यादा इजिली जाएंगे क्योंकि उसकी सेल मेम्ब्रेन के साथ जो फॉस्फोलिपिड्स हैं नेगेटिवली चार्ज, उनके साथ वो ज्यादा अच्छे इंटरेक्ट कर सकते हैं। तो अगर ऐसे सेल्स होंगे हमारे पास जो कि इनके साथ इंटरेक्ट कर सकते हैं, तो उनके अंदर वो उसके साथ इंटरेक्ट करके वो आराम से अंदर इंटरलाइज कर देते हैं। इसके पीछे भी मैकेनिज्म होती हैं, 3-4 मैकेनिज्म होते हैं, आप लिटरेचर में जाके पढ़ भी सकते हो कि व्हाट आर द मैकेनिज्म्स फॉर इंटरलाइजिंग इनटू द बैक्टीरियल सेल्स। तो वो या तो डिफ्यूज कर जाते हैं या वो इलेक्ट्रोस्टैटिक इंटरेक्शन करते हैं, बट वो स्पेसिफिकली वहां पे इंटरेक्ट कर सकते हैं। तो मैंने क्या किया, मैंने ये किया कि वी हैव ट्राइड टू मेक अ टारगेटेड सिस्टम। अब ऐसा सिस्टम बनाया जो कि सिर्फ बैक्टीरियल सेल के साथ ही इंटरेक्ट करेगा, ह्यूमन सेल के साथ इंटरेक्ट नहीं करेगा। सो दैट वाज द अंडरलाइन। पर उसका रीजन सिर्फ ये है कि जो पेप्टाइड विच वी हैव यूज्ड, that peptide is specific for bacterial cell because of the difference in the composition of the bacterial cell membrane and the human cell membrane. So I hope मैं आपको समझाने में I have been successful कि the composition of bacterial or the microbial cell membrane is different from the human cell membrane, and that is why some of the peptides are specific for bacteria and not for human, और उनको ही हम नैनो मटेरियल के साथ लगा के हम उसको सेल के अंदर इंटरलाइज करवा सकते हैं। डॉक्टर शेरी हैज आल्सो वर्कड अपॉन दिस, शी हैज वर्कड अपॉन दिस सेल पेनिट्रेटिंग पेप्टाइड, शी कैन आल्सो मेक यू अंडरस्टैंड दिस प्रॉब्ली। यस, नेक्स्ट
Any
क्वेश्चन।
other question you can ask? You can ask directly from sir. If you don't want to write in the chat box, you can ask directly from sir
Yes, you can
by
unmute and
unmuting your audio.
Sir, एक question, one more question.
yes,
Please tell about FITC labeling.
ओके। तो अब मैंने मान लीजिए जैसा गोल्ड नैनो पार्टिकल बना लिया, ये वाला गोल्ड नैनो पार्टिकल बन गया, सॉरी, ठीक है, उसके साथ मैंने सेल पेनिट्रेटिंग पेप्टाइड भी लगा दिया, सब कुछ अच्छा हो गया, वेरी गुड। बट जब मैं इसको, तो मुझे कैसे पता लगेगा ये कहाँ गया? मुझे तो पता ही नहीं लगेगा क्योंकि मैं तो इसको चेक ही नहीं सकता, क्योंकि इसमें से कुछ भी ऐसा नहीं है जो लाइट इमिट करता है या जो मैं चेक कर सकता हूँ। जैसे आपने एमआरआई स्कैन कराते हो, सीटी स्कैन कराते हो ना, उसमें भी फ्लोरोफोर डालते हैं, वो चेक करता है बॉडी, जैसे जैसे आप जाते हो वो लाइट इमिट करता है। तो उसके लिए एफआईटीसी जो होता है उसे कहते हैं फ्लोरोफोर। फ्लोरोफोर वो होता है जो फ्लोरेसेंस शो करता है। फ्लोरेसेंस आपने पढ़ा होगा फिजिकल केमिस्ट्री में भी, वो एक तरह का मैकेनिज्म है फ्लोरेसेंस, जिसमें कि लाइट जो है वो एमिट होती है अगर एनर्जी लेवल्स के साथ एनर्जी ट्रांसफर करती है। वो आपने जेब्लॉक्स की डायग्राम पढ़ी होगी उसके हिसाब से। तो कुछ मॉलिक्यूल ऐसे होते हैं जो फ्लोरोफोर्स होते हैं, जो फ्लोरेसेंस शो करते हैं। तो हम क्या करते हैं, जब हमने कोई कॉन्जुगेट सिस्टम बनाया तो हम उसके साथ कुछ ऐसे मॉलिक्यूल लगा देते हैं, जैसे कि FITC। FITC जो है वो फ्लोरो आइसोथायोसाइनेट ग्रुप है, इसका पूरा नाम आप गूगल में भी चेक कर सकते हो। तो वो जो है वो बेसिकली एक केमिकल स्ट्रक्चर है, केमिकल ऑर्गेनिक मॉलिक्यूल ही है, जो कि लगता है और वो फ्लोरेसेंस शो करता है। तो जैसे ही मैंने अपने कॉन्जुगेट के साथ फ्लोरेसेंस लगाया, फ्लोरोफोर लगाया, तो उसकी फ्लोरेसेंस आनी शुरू हो जाएगी। अब जैसे ही मैंने इसको सेल में डाला तो मुझे इस तरह के सिग्नल दिखेंगे, ये दिख रहे हैं ना वो ग्रीन सिग्नल, ये ग्रीन सिग्नल फ्लोरोफोर की वजह से दिख रहे हैं। तो अगर ये फ्लोरोफोर ना होता तो मुझे सिग्नल नहीं दिखने थे। तो चेक करने के लिए कि वो बायो सिस्टम के या सेल के अंदर या बॉडी के अंदर गए हैं, तो फिर मैं उसको ये माइक्रोस्कोप के अंदर डाला है, फ्लोरेसेंस माइक्रोस्कोप के अंदर चेक कर रहे हैं उसको। तो फ्लोरेसेंस माइक्रोस्कोप जैसे ही करते हो तो उसकी फ्लोरेसेंस ऑन हो जाती है, तो आपको सिग्नल दिख जाते हैं। तो फ्लोरोफोर का काम ये है कि वी आर जस्ट ट्राइंग टू सी वेदर दैट पर्टिकुलर एंटिटी, जैसे कि हमने ये बनाया गोल्ड नैनो पार्टिकल सेल पेनिट्रेटिंग पेप्टाइड सिस्टम, ये हमारी बॉडी के अंदर कहाँ गया और कैसे जा रहा है, मतलब क्या उसका रूट है, सब कुछ आप चेक कर सकते हो अगर फ्लोरेसेंस माइक्रोस्कोपी हो। यही फिनोमिना CT स्कैन, MRI और इन चीज़ों में भी होता है कि उसमें भी यू कैन चेक, बट उसमें स्लाइटली डिफरेंट होता है। उसमें वाटर मॉलिक्यूल्स जो हैं, MRI तो NMR बेस्ड है, वो थोड़ा डिफरेंट है, बट फिनोमिना ऑलमोस्ट सेम ही है इससे। यस, आई
Sir,
होप दिस
next
इज
question. Yes, sir. Sir, next question is how colors play a role in nanotechnology?
Oh, that's a very actually good question. So, अब क्या है, व्हाट इज कलर? कलर कहाँ से आ रहा है? कलर इज कमिंग बिकॉज ऑफ डिफरेंट वेवलेंथ्स। ठीक है, जैसे ये दिख रही है आपको ना यूवी की वेवलेंथ, ये वेवलेंथ जो है ये नैनो पार्टिकल दिखा रहा है 500 समथिंग पे, 550 से थोड़ा कम, 540 520 पे। और आपको पता है कि विजिबल रीजन क्या होता है? विजिबल रीजन होता है 400 से लेकर 800 नैनोमीटर तक। 400 से लेकर 800 नैनोमीटर तक आपके पास सारे कलर आ जाते हैं। यू नो ना, आपको पता है ना कि 400 पे वॉयलेट कलर होता है, 800 पे रेड कलर होता है, वो डिफरेंट कलर्स आते हैं, एंड आप वो आप उस हिसाब से देख सकते हो। तो आपकी आई सिर्फ वही वाला रेंज देख सकती है, 400 से 800 वाला। सो ये नैनो पार्टिकल का बड़ा अच्छा सिस्टम है। कैसा सिस्टम है कि अगर जैसे ये गोल्ड नैनो पार्टिकल बना, ये पर्टिकुलर वेवलेंथ दिखा रहा है 500 समथिंग पे, 500, वो जो भी कलर है वो रेड कलर को कॉरस्पॉन्ड कर रहा है। ठीक है, एक्चुअली मैंने उल्टा बोल दिया, 400 वाला रीजन जो है वो रेड का है, 800 वाला रीजन जो है वो वायलेट का है। सो वो जो है रेड कलर को शो कर रहा है, तो इसका रेड कलर होगा। लेकिन जैसे ही आप इसे एग्रीगेट कर दोगे उसकी वेवलेंथ चेंज हो जाएगी। वो वेवलेंथ आप देख रहे हो, ये वाली पर्पल वाली ये इधर चले गई, तो वो जैसे ही वो रेड कलर वो गायब हो गया। सो नैनो पार्टिकल्स या नैनो केमिस्ट्री के अंदर हमें कलर के साथ कोरिलेशन बहुत अच्छा मिल गया, इस तरह से कि इनकी जो वेवलेंथ जनरली है वो कलर्ड सिस्टम में आती है। और जैसे ही आप उसको चेंज करते हो, वंस यू चेंज सम पैरामीटर, जैसे कि हमने इंटरेक्शन चेंज कर दिया, एग्रीगेट कर दिया या कुछ और चेंज कर दिया, तो उसकी वेवलेंथ चेंज कर जाती है। वो वेवलेंथ चेंज करती है तो कलर चेंज हो जाता है। तो उस कलर को हम अप्लाई कर रहे हैं अपनी सिस्टम से। So actually it's a very good question, a very basic question, a very nicely put question. So इस तरह से आप correlate कर सकते हो simple phenomena that you are actually changing the wavelength by changing the parameters, and that you can see from your eye that color is coming. So this is the basics of this application. I hope this is understandable. Yes.
Thank you, sir.
Thank you so much, sir. On behalf of Chemistry Department, DAV College, Abohar, my sincere thanks to our guest, Dr. Rohit Kumar Sharma, for being with us today. Thank you, sir, for gracing your important work and sharing with us your findings and opinions, excellent presentations, and making this webinar interesting and meaningful. The use of nanomaterials in various fields, especially in the coronavirus vaccine, is incredible. I would like to express profound gratitude to our principal, sir, Dr. Rajesh Kumar Mahajan. I would also like to thank you, HOD Dr. Suresh Kumar Sharma, for his moral support and guidance. Thanks to our staff members who have made this webinar a grand success. Finally, the wonderful students for your cooperation. Once again, I thank all for your cordial cooperation. Thank you.
Thank you very much for giving this opportunity, And thanks to your chairperson, thanks to Dr. Suresh, and thanks to Dr. Sherry for having me here. And in case you have any query, you can take my email ID and you can write an email to me anytime. Thank you very much.
Thank you very much, sir. Thank you.
Thank you, sir.